Right. Hello, my name is Octavio Marin-Pardo, and first of all, I would like to thank the organizers for the opportunity to share this presentation, which is use of a low-cost electromyography biofeedback system for telerehabilitation in chronic stroke. Research has shown that high-dose interventions of repeated movement in the order of hundreds or thousands of repetitions can promote chronic stroke upper limb recovery. However, standard clinical practice provides limited dosages, primarily due to time, physical, and economic constraints. At-home telerehabilitation services, supervised by a clinician, are a potential solution to provide higher dose interventions. Measurement and feedback of muscle electrical activity from the impaired hand via electromyography can be incorporated into telerehabilitation interventions. Specifically, attempting to reduce unintended co-contractions might be beneficial for improving motor control. These may be particularly useful for people with moderate to severe impairment, that is, without active movement. Therefore, we developed a low-cost system called TeleReinvent to provide at-home repeated practice with telerehabilitation and EMG biofeedback technology. And today, I will highlight the results of a pilot test we performed to evaluate the feasibility and safety of this system. We recently published the details of the development of the system and the feasibility test, so if you're interested, you can see them here. This system essentially consists of low-cost EMG sensors that use standard EMG electrodes and connects via USB to a laptop computer. Our software is a collection of modules for acquisition, processing, and interaction. Here, we can see examples of the graphical interfaces that show relevant information and configurations depending on the user's role of as a participant or a clinician. Here we can see screenshots of 3 different feedback games that we developed for enhanced motivation. In Skibble, appropriate muscle activity is translated as different scores. In Blinko, the participants use their muscles to move the character and then drop a coin. Lastly, in CEMG, signals are mapped to a color scale or shown as a single trace along with the session maximum. You can scan this QR code for an immersive demo of these games and other projects of the USC Smart VR Center. To interact with the games, we take the muscle signals and calculate a ratio of their activity. Since we are interested in training wrist extension movements, values that are closer to isolated extension are mapped into higher scores, triggering the corresponding animation in the game. Here we can see the system in action. As I mentioned before, muscle activity is translated as movement of this arm to push the ball towards the board. For this pilot test, we recruited 1 chronic stroke survivor with severe impairment and asked them to play with the skee-ball game. On each session, videos show how to place the sensor and calibrate the system. Then the game starts. Each trial consisted of 7 seconds of rest followed by 5 seconds of movement attempt. A total of 100 trials per session were recorded for 40 sessions over 10 weeks of training. As we can see here, after 40 training sessions, there was a significant increase of individual activity over time. In this plot, each value represents the average of the 100 trials of each session. In conclusion, this pilot test suggests that the system is feasible, safe, and enjoyable to use during 10 weeks of biofeedback training. Low-cost technology for individuated muscle training may be feasible to reduce unintended co-activation during home-based telerehabilitation. However, it is important to note that due to the safety protocols during the COVID-19 pandemic at the time of this test, we were not able to perform clinical assessments. So further research is required with a large sample size to examine the efficacy of this technology to provide personalized interventions and promote chronic stroke recovery. Finally, I would like to thank the Neuroplasticity and Neurorehabilitation Laboratory, the USC Smart VR Center, and all the agencies that funded this work. Thank you very much.